The overall goal of the following experiment is to visualize the migration of dendritic cells into the draining lymph node using non-invasive fluorine magnetic resonance imaging, or MRI, in combination with conventional proton MRI. This is achieved by first preparing murine bone marrow-derived dendritic cells and labeling the fully differentiated cells with fluorine-rich particles. During the second step, the fluorine-labeled cells are administered intracutaneously into the hind limbs of a mouse, ensuring the efficient transmigration of the administered cells. Next, the anatomical region of interest is imaged by fluorine proton MRI to monitor the migration of the dendritic cells from the skin to the draining lymph nodes. Ultimately, dendritic cell migration from the area of application into the draining lymph node can be visualized by fluorine MRI.
The main advantage of this method over other cell tracking techniques, such as those requiring iron oxide nanoparticles, is that carbon-bound fluorine is virtually absent in all living organisms. This yields complete background-free images and a complete cell selectivity following application of fluorine-labeled cells in vivo. This method can help answer questions in the field of immunology, such as what is the influence of molecular candidates in the migration of dendritic cells in vivo. Demonstrating the method is Julia Skłodowski, a technician, and Helma Vaikses, a senior scientist from my laboratory.
On day 10 of bone marrow dendritic cell culture, activate the dendritic cells with LPS for 24 hours. Label the dendritic cells with